Welcome everyone to this week's edition of the Extracellular Vesicle Club. Today we're having an event with Ferdinando Pucci at the Oregon Health and Science University in Portland, Oregon. And he's going to be telling us a little bit about cancer stem cells and their niche and how EVs and macrophages and T cells are involved there. So I'd just like to welcome everybody. While we are listening to the presentation, I'm going to have everybody on mute. But please put your questions and your comments in the chat box. And then at the end of the presentation, we will get to you and have a robust discussion. So Ferdinando, thank you so much for joining. We're really looking forward to hearing about your work.
Yeah, thanks, Ken, for inviting me. It's my pleasure to go over this recent paper that we had. And thanks everyone also for joining today. As Ken mentioned, I will discuss a bit the work we've done to study the role of extracellular vesicles in the cancer stem cell niche. And as you can already see, we found that macrophages and T cells are also part of this very specialized tumor microenvironment. So my name is Ferdinando Pucci. I'm in Portland, Oregon at Oregon Health and Science University. And if you are one of those social media people, please follow our lab and our work on Twitter. So a brief outline of what I will discuss today. I will start with the problem. How can we study extracellular vesicles from rare cell populations, such as cancer stem cells, and why that is important? Our solution that we developed and I will describe today is what we call EV painting, which technically is cell surface labeling via extracellular vesicles. This technology is based on an enzyme called sortase A, and I will describe its properties and how we use it. And the application here, again, is in cancer stem cell and how cancer stem cells interact with other cell type host cells in their niche. Finally, I will also mention, of course, some limitations of our study that will open also the way for future experiments and directions. So I'm not by any means a cancer stem cell expert, but quickly looking in the literature, you can immediately understand how the field of cancer stem cell is important, and many people are working on trying to figure out how these specialized stem-like tumor cells can communicate with the host cells, not only of the immune system, but also other stromal cell types. Cancer stem cells are also believed to communicate with differentiated cancer cells. And this communication is generally bidirectional. So they receive signals and they can send signals. What are these signals? These signals, the most classically studied signals are, of course, cytokines, growth factors, and other extracellular secreted factors such as proteases and extracellular matrix components. Recently, extracellular vesicles have been included in this range of factors that mediate communication between cancer stem cells and its environment. And this is important because it has been shown that cancer stem cells, of course, can sustain tumor growth and can provide a mean for tumor escape and recurrence after therapy. And so understanding how these cancer stem cells communicate and sustain these tumor niche is important to then identify novel therapeutic targets that can be developed and assessed in preclinical models. So let's get to the problem of how do we understand the communication between the cancer stem cells and their niche cells, in particular the one that is mediated by EVs. So one solution So initially we thought there are two solutions to this problem. One could be that we perform a large scale isolation of these rare cell subsets. We put them in culture if culture conditions are available, and then with these large-scale cell supernatants, we could purify the extracellular vesicles for in vitro characterization, and then functional assays either in vitro or in mice, in vivo. The other solution that we thought was we could use genetic reporters of EVs, and once these are validated, we could then express these genetic reporters within the rare cell subset, either in vitro or in vivo, and then perform functional studies this way. And we put our money on the second solution because not only that is our expertise in generating and validating genetic reporters, but also because there are this avoids a number of biases that are encountered when there is the need of in vitro purify the EVs and perform further manipulation for the in vivo transfer. The problem still is also that we don't know exactly, how much genetic reporter or even a non-genetic reporter EVs can carry to then allow us to identify their target cell. And this is because, as you all know, if the cell was the size of the US, then an extracellular vesicle would be the size of a Portland metro area. They are very small, so they can, of course, carry very little reporters. And so we developed in the past a number of genetic tools to track native tumor-derived extracellular vesicles at different scale. We included luciferase to track the biodistribution at the whole body level, all the way down to Curie recombinase to understand the molecular mechanism of cell-EV interactions. In this case, we picked this reporter that allows us to track the EVs and their cellular targets. This is the CD63-GFP fusion protein, which also expresses a truncated receptor delta LNGFR. This is just, a protein that is used, is a transmembrane protein that is also included in the EVs, and it's used for labeling the EVs with antibodies. And so this is what we call first-degree labeling, because the reporter is in the EVs, and then we use that reporter to then identify the host cell. In this case, again, the reporter is a genetic reporter, so we engineer the tumor cells in vitro, then we implant these tumor cells in mice, and then we study, after the tumors outgrow, we study what happens and where the reporter signal is found. This system works very well, although the signal, as I mentioned, is quite low. Here you can see an image cytometry analysis of a tumor macrophage from these tumors. And as you may know, tumor macrophages are among the cell types that engulf the highest amount of EVs, and still this is the signal that you get. So it's very dim. If you go away from the tumor in the lymph nodes, where most of the EVs naturally drain, you can see that the signal becomes even dimmer. Here you see B cells gated for the surface reporter, NGFR in this case, and you can see that only less than 1% of the B cells here are detected to be binding the EVs. But we know that actually the vast majority of the B cells now, we know that they are actually interacting with the EVs. So first-degree labeling can be not enough to study, especially long-distance communication via EVs. And so this is why we developed this system called EV painting that is based on the use of an enzyme, this sortase A. Sortase A is a bacterial transpeptidase that is, again, engineered as a transmembrane protein. And this transmembrane protein is then expressed in your target cell of interest. This enzyme generates a covalent bond between a fluorescent substrate that is detected via a consensus sequence, which is LPET-G, and a protein with an N-terminal glycine. And on the surface of cells, there are several N-terminal glycine-containing proteins that are pretty much ubiquitous, like class 1, several integrins, class 2, and other adhesion molecules. And so we thought, why don't we do, instead of a first-degree labeling, we could do a second-degree labeling in which we're not labeling directly the EVs to identify the target cells, but we could take advantage of the much larger cell surface of the target cell to carry the fluorescent reporter. And in a process that requires the binding of the EV. And so in this way, we hope to improve the sensitivity of detection of the EV targeted cells, even in distant organs. So let's quickly go through how we validated this system, because it will be important to understand before we get into the cancer stem cell work. So to validate a genetic reporter of EVs, we started by designing different reporter configurations. Here, for example, the sortase A, there are many publications in the literature about it, and there are many variants that contain a different number of mutations. Here we picked the two different, two of the most recent mutations, one that contains the 3 different amino acids that has been evolved for higher processivity and efficiency, and another one that contained 11 mutations in its sequence. The 3 mutation version was used in an in vivo study recently to study the interaction between the dendritic cells and the CD4 T cells in vivo. So we thought it was a good candidate for these studies. These 2 variants were designed to be expressed in 2 different configurations. One was a short configuration fused to a simple transmembrane domain. And the other one was actually fused to what we used before as a reporter, the delta LNGFR. And this was because we wanted to try to have the enzyme farther away from the surface of the EV, hoping that maybe would have helped in the conjugation of the fluorescent reporter to the target cell. But as you can see here, when we generated the vectors and expressed these sortase designs in cells, only one was actually expressing at very high levels. And so this is the one that we picked, the 3 mutation shorter transmembrane domain. It's important to note that this is done using 1 copy of a vector per genome, because in this way, you can actually compare the expression level. And most likely, the other designs just do not express because of folding issues or aggregation issues within the cell, and they are immediately degraded. So this is a way to start identifying which is well expressed. Then, of course, we have to assess the function. And accordingly, when you add a substrate, biotinylated substrate, the same construct that we selected was the one that was actually the top performer. We then assessed whether this reporter was going to change the size of the EVs, their morphology, or even some of their markers. And we found no difference in control EVs and those that actually carry the sortase by both electron microscopy and nanoparticle tracking analysis. And about the surface markers, we still observed that they are fully expressing CD63 and they carry the sortase on the outside layer, surface of the EV. These are all surface staining, basically, of single EV by limiting dilution. And we also used, as you can see here, NGFR has a pan EV marker. Yeah, all the EV prep that was done by ultracentrifugation in this case is uniformly expressing these three markers. An important part was to assess whether the presence of the sortase on the EVs was inducing binding to a target cell. This, of course, is not good, because then we are having a false positive result. And so we compared a control EV with an EV that contains the sortase A, and we assessed the level of NGFR on target cells. We incubated these target cells with these two different EVs at different dilutions, and we observed that the staining with the reporter NGFR, which is independent from the GFP or the sortase, the staining with the NGFR was similar, whether the sortase was present or not, which indicates that the sortase is not driving the binding via its affinity for the substrate. And indeed, this is in agreement with the enzymatic constant, which is a measure of affinity of the enzyme for the substrate, which is in the millimolar range. So it's still very high, low affinity. Importantly, when we assessed several different EV sources for binding and labeling of a target cell, in this case, the target cell was an endothelial cell line, we found that as compared to the GFP reporter, the CD63 GFP reporter, the presence of the sortase allows us a tenfold increase in detection sensitivity, which is exactly what we wanted to study, to use this system in vivo to identify EV target cells. And so, brief summary, genetic reporters are an unbiased and convenient way to study EVs directly in vivo from rare cell subsets, including rare cell subsets. First-degree labeling can be not sensitive enough in some situations, as we have seen, especially to study long-range communications, to identify EV-mediated impacts on target cells. Self-surface labeling of EV target cells can improve the detection of these host cells by tenfold. And so let's see how this system works for this application on cancer stem cells. So one of the advantages of using genetic reporters is that you can decide where the reporter is and how the reporter is expressed. In this case, we use the tissue-specific promoter that is restricted to cancer stem cells. We actually use the two tissue-specific promoters, ALDH1A1 and a SMAD responsive element that drives the expression of the sortase, the transmembrane sortase. And so in this way, the sortase is expressed only in cancer stem cells. As a control, an internal control, we used a constitutive promoter, here in orange, that drives the expression of the first-degree marker CD63 GFP, which is now expressed by all tumor cells, but also a fluorescent substrate, which is a red fluorescent protein called M-scarlet, fused to the LPET-G consensus sequence for the sortase, which means that the sortase substrate is produced by all tumor cells and is released in the extracellular environment like a cytokine. It is fused to a signal sequence, and so it is secreted in the tumor microenvironment. As a control group, here we only have the presence of the substrate and not the presence of the sortase. And so this allows us to detect the differential increase in substrate conjugation in the absence of the sortase, absence and presence of the sortase. So this system worked pretty well. The promoters were specifically targeting cancer stem cells. You can see on top in vitro culture conditions, you can see that the ubiquitous promoter PGK labels all the tumor cells. The tumor cells here are MIR, which is a type of squamous cell carcinoma. If the CD63 GFP is expressed under the cancer stem cell specific promoter ALDH, then you have many less cells that are expressing it, suggesting that those could be bona fide cancer stem cells. On the bottom panels, you see the same cell lines in vivo, a different resolution from the whole tumor section down to the cellular level. And again, you can see that the expression of the CD63 GFP by the cancer stem cell specific promoter is very much restricted. When you zoom in, you already see that the few GFP positive cancer stem cells may have a particular distribution as compared to macrophages and the total of immune cells. Whereas the ubiquitous promoter, of course, labels all the tumor cells, both differentiated and stem cells. We did some functional validation of these reporters. Of course, we flow sorted the top expressing tumor cells for this GFP reporter, and we performed culture conditions in non-attaching media or vessels. In non-attaching media, these cells, if they are cancer stem cells, would form orospheres, and we performed gene expression analysis of these orospheres and compared to the bulk attached cell cultures. And we found that many stem cell markers, bona fide stem cell markers, are highly expressed in the orospheres, including the GFP. And this is a quantification of how many orospheres are formed by the cells sorted as ALDH positive or negative. You can see that the negative basically do not form orospheres and they just die out because they cannot attach. Okay, so we have a good reporter to express an EV marker genetic reporter of EVs in cancer stem cells. And so we first started using the first degree tracking system of cancer stem cell EVs. Here you have a description of a schematic of the experimental groups. So we have the squamous carcinoma cell MIR untransduced. We have the bulk of the MIR population that expresses the ubiquitous reporter CD63 GFP. So thereby here the EVs from both stem and differentiated cells are labeled. And then we have the reporter that is specifically expressed in cancer stem cells using both these two reporters in different mice, but separately. We employed both of them. Both the promoters, the LDH and the SRE. And so this is what we found at endpoint, which is about 2 to 3 weeks after tumor challenge. We collected the tumors and we performed the flow cytometric analysis of the immune cells and some stromal cells and assessed the level of GFP that was on each of these immune cell subset or host cell subset. As you can see, in the case of untransduced GFP negative tumor cells, we barely have any signal. In the case of total EV labeling using a constitutive promoter, we have, as expected, macrophages that are highly labeled by these endothelial cells, neutrophils and monocytes that are the most, and also B cells, but B cells are very few in the tumors. In the case of restricting the reporter expression into the cancer stem cell, then the situation changes a lot for most of these subsets. However, we observed that in specific subset, the class II negative macrophages and the PD-1 positive T cells, there was not a lot of change. So in this case, we were interested in the absence of a difference, of statistical difference. And this is because we reasoned that if labeling all tumor EVs versus labeling cancer stem cell EVs does not give any difference, that means that these cells, most of the time, hang out in the cancer stem cell niche. And this was actually what was also exemplified in this specificity index that we calculated based on these values. These two cell types had a high cancer stem cell niche specificity index. We then went to check the same outcome because we weren't really sure if to trust this first approach with the EV painting approach, hoping that being much more sensitive, he would have highlighted whether we were seeing the right thing or not. And in this case, again, as I mentioned before, we have these two groups, a test group that contains the LDH with the sortase expression, and a control group that only has the expression of the fluorescent reporter, sorry, the fluorescent substrate for the sortase. The internal control, again, has the first degree labeling, the CD63 GFP, in all the tumor cells, including cancer stem cells. And so the calculation of the EV painted target cell was the M scarlet positive minus the control M scarlet positive, because you could argue that since the fluorescent substrate is secreted, it could be picked up by host microenvironmental cells, specifically by simple panocytosis. And so, to our delight, it actually confirmed the same results of the first degree labeling, showing that, again, there were the same two different cell types, the class II negative macrophages and the PD-1 positive T cells, that were not, that showed no difference between the CD63 GFP first degree labeling and the EV painted approach, suggesting again that these two cell types are likely to be present within the cancer stem cell niche. So in this case, we also quantified the distance between, oh, sorry, yes, this is the slide. The genetic reporter for EVs allows to express a reporter cassette that can synergize to detect the EV and control the labeling of EVs in vivo. The cancer stem cell EVs target a specific subset of tumor-infiltrating immune cells that are class II negative macrophages, which are generally also known as alternatively activated macrophages, and PD-1 positive T cells. So what could be the limitations of this study? One limitation is that although the two approaches that we used to label the EV target cells for cancer stem cells were in complete agreement, we cannot really distinguish between cell-cell interactions and EV cell contacts in this case, especially because we are studying the same location where the rare cell subset is found, in this case, the cancer stem cell niche. And this is basically due to the fact that, as far as I know, but I'm happy to learn from you if you know ways to genetically label the EVs, but not the parental cell. Another limitation is that it's not clear if in this case we are really targeting all the EV subsets that are known, large EVs, small EVs, annexin positive EVs or ectosomes like the AR-DC1 positive EVs. We performed a limited marker analysis on our EVs. Then functionally, biologically, we didn't study whether it's the cancer stem cells that induce the macrophage phenotype or the tumor or the T cell phenotype, or if it is vice versa. If it's the presence of those macrophages that induce tumor cells to revert into cancer stem cell phenotype. And this, of course, is an important distinction. And last, we didn't perform any profiling of class II negative macrophages and PD-1 positive T cells. These would have helped understand further what is the impact of either cell-cell contact or cancer stem cell EV contact in this situation. And I'm going to conclude here by acknowledging the work of Patricia, who did a great job with the cancer stem cell in vivo study. And Nick was the main author for the in vitro validation of the sorties. And I would like to thank current and past funding and you all for attending this journal club. And if you have any question, I'm happy to try address them.
Very good. Well, thank you so much, Ferdinando. And I've seen some new people coming in since you started. So I just want to remind everyone that if you do have questions or comments, please place them in the chat box. And I'm actually going to go to the controls right now and allow people to unmute themselves. So when I call, when you put your question in, I will call on you. And then you can have a conversation with Ferdinando. Maybe I can get started with a question about how you can use this system. Like, do you think that you can use this system to understand what's happening in the human?
Good question. I don't think so. I mean, unless you have a humanized mouse model, you probably cannot use these reporters in patients, right? Not even for you know, biodistribution studies.
Yeah.
So, these are all foreign proteins.
Sure.
Bacterial proteins, GFP. So you would have an immune response. You would basically immunize the patients. I don't think any agency would allow that. Yeah.
So we'd basically be working with something that we would take ex vivo then, if we want to use this system. Yeah,
this system is purely for understanding the biology of EV in preclinical models. You could have a humanized mouse that has patient-derived xenografts, and those can be engineered to express these reporters. Then you will have the interaction with the human cells in the human immune cells in the mouse. I think that, or other in vitro studies, but with the limitations of in vitro studies, of course.
And so then you would not anticipate that the enzyme could be used, for example, to release a surface cargo or something near a cell. Maybe there'd be some kind of triggering event, and that's because of the immune response.
Yeah, I think it would not be feasible because of the immune response.
Okay. Yeah.
Unless, you know, there are ways to avoid the immune response to xenoantigens by detargeting their expression from antigen-presenting cells. But in this case, you would have to detarget the binding of the EVs from, like having something like, I don't know, CD47, a don't eat me signal or something like that, to avoid internalization and processing of these potentially therapeutic IVs into antigen-presenting cells in patients. I guess it could be a possibility, yeah.
Good, all right. Okay, let's see. Do we have any questions from the audience? So we have a question from N. Ali. Please go ahead.
Hi, is ALDH1 a good biomarker for cancer stem cells derived from any pan cancer, or is it specific to one type of cancer?
Yeah, that's a great question. As far as I understand, there are a few different cancer types that whose cancer stem cells are overexpressing this ALDH. The squamous carcinoma was one, but I know Patricia was working also with mammary carcinoma using the same promoter. So at least these two cancer types, cancer stem cell types, express ALDH, and so that can be used.
So, sorry, it's a related question. If you have a cancer, like cancer stem cells isolated from patient based on the biomarker ALDH, will that also represent from the serum, like the position of ALDH marker inside from the serum? So if CSCs are increased, or CSCs have ALDH1 positive and it's very high, when you isolate EVs from them, will it correlate with the total EVs isolated from the serum? Not from just CSC.
So you're asking whether the circulating EVs mainly derive from cancer stem cells?
Yeah. I
don't know. I would think that would be unlikely, because cancer stem cells are still a rare subset of the bulk tumor mass, right? And so possibly most of their EVs would be trapped within the niche. We haven't looked into the draining nodes specifically in this case, but if you don't find them there first, I would doubt they would even reach the systemic circulation, because first they go through the lymphatics, which filter the EVs. So you really need to have a spillover, either a high release or a spillover back into the circulation directly from the cancer stem cell niche. But yeah, I may be wrong.
Okay, thank you. Can I ask one more question?
Yeah, of course.
Go ahead.
So for ALDH1, like as a biomarker, do you have any other markers pertinent to cancer stem cell other than ALDH? Yeah,
we used a different promoter. So just to clarify, this ALDH is the sequence of a promoter, right? And we use that to drive the expression of the EV reporter, specifically in cancer stem cells. So we used a second promoter, which contains sequences for from the SMAD responsive element. And that is because SMAD signaling and TGF-beta signaling has been shown to be really important in cancer stem cells. And so my colleague in the department, actually, Dr. Naoki Oshimori, was the one who provided that promoter. And he's been using that a lot for, again, for squamous carcinoma, even genetic models of squamous carcinoma. So that is also another promoter to target cancer stem cells. Yeah.
Okay. Thank you so much. Those are my questions.
Yeah. You're welcome.
Thank you. Thank you. We have another question from Lothar Dietrich.
Yeah, hi, Ferdinando. Nice talk.
Hi.
Thanks. Interesting approach.
Thank you.
I actually have, before I start the question, I typed there. I have a very basic question. I'm not quite sure if I understood exactly how you targeted the sortase to the EV. So this was a fusion protein with the EV marker, and is that then present in all EV subsets, or is that enriched in small EVs or large EVs? What you did now?
We simply fused it to a transmembrane domain.
Okay.
It's just a simple transmembrane domain, which is, I think it's from PDGF receptor, if I remember well.
I see. So you did not attempt to target it now to any specific EV population, or?
No, we wanted to be as broad as possible to start with, right? Because we didn't want to bias ourselves to begin with. But definitely it could be targeted by fusing specific markers such as CD63, Alexin A1, or other markers that you may be interested in. We did two designs. One was with a transmembrane domain, a simple transmembrane domain, of course, lacking the intracellular signaling domain, and the sortase fused with a short spacer right outside, and another design that had a transmembrane domain with the rest of the receptor, the NGFR, which is a quite long protein that we thought having the sortase at the tip would allow it more flexibility and more reach toward the target. But that didn't really work very well. It was not expressed
at decent
levels.
Yeah. Okay, and then my other question, actually, I was thinking now about these two cell populations. I mean, you showed several images where you can clearly see these cancer stem cells close to macrophages, so I guess that makes sense. But what about the PD-1 positive T cells? Have you tried to look at them in the tissue? Can you see them also now in physical proximity, or are you rather suggesting this is a very specific uptake mechanism that can also happen at a distance, perhaps?
Yeah, it's a really good question. We didn't check for the T cells in this case. That's unfortunate. But yeah, it would be very interesting to see whether there is actual colocalization within the niche of PD-1 positive T cells, especially because I understand that cancer stem cells have very low levels of class 1. So that could again induce some energy into the T cells in their vicinity, possibly. So yeah, that's also another limitation of the study. We didn't do that.
All right. Thank you.
Thank you.
Thank you. Well, it looks like we've come to the end of the questions in the chat box. If anybody has a last-minute comment or question, please put it in or raise your hand. But if not, then thank you all for joining today. And special thanks to Ferdinando. Thank you so much for sharing your work with us. It's very exciting, and we'll look forward to seeing how you continue to use this system in the future. So
thank you.
All the best, everyone. Take care, and we hope to see you again soon.